Today, this is the second guest lecture for the month of November, and we have Professor Chamindie Punyadeera from head of head saliva and liquid biopsy translational biology translational laboratory, Griffith Institute for Drug Discovery, Griffith University, Australia. Let me take a minute to give a brief introduction to her. She's Professor Chamindie is an inventor and an ambassador to women in STEM. She has had a hybrid research career working in industry as well as in academia. She leads this saliva and liquid biopsy translational biology translational laboratory at Griffith University in Brisbane, and her laboratory develops biomarkers for concept through to commercialization, and she has made groundbreaking discoveries and has led to the world's first detection of an occult HPV-driven oropharyngeal cancer in a healthy person, laying the foundation for a screening trial. Her research into saliva-based tests to early diagnose and predict head and neck cancer received FDA approval under breakthrough device designation for her industry partner in 2021. In addition, her laboratory develops and applies liquid biopsy-based technologies to early detection and predict outcomes in head and neck cancer, lung cancer, and glioblastoma. So she has authored more than 150 papers and also achieved several grants, and she's also a grant reviewer for both national and international funding agencies, and also member of editorial board for the Journal of Oral Oncology, associate editor for BMC Genomics, and a guest editor to BMC Medical Genomics, Diagnostics, and Biomolecules. So for today, she's going to talk about the application of liquid biopsy to cancer and salivary diagnosis. So the floor is over to you, Professor Chamindie.
Good morning, and thanks, Dulmani, for the introduction. And can you see my screen?
Yes, we can see it.
You can?
Yes.
I'll be giving you some examples where we have used saliva as a next generation diagnostic fluid to early detect heart failure and cancers. So mainly head and neck cancers, and to predict outcomes in glioblastoma or brain tumors. And we are also applying liquid biopsy. So by definition, liquid biopsy is the use of biomarkers present in body fluids. We all know when a person is diagnosed currently, it's through tumor tissue, but the tumor tissue is very heterogeneous. And more so, when a patient has had surgery, tumor is removed. So how do you track the response to treatment in a patient? And that's through biomarkers present in both saliva and blood. Can you really see my screen now? Because I don't see that highlighted. You can, right? Can you see it?
Yes, we can see it very well.
Oh, okay. So this is
is muted. That's the reason you don't hear her.
Okay. This is my disclosure statement highlighting funding and patents, and I'm also a co-founder and a director of a startup company in Australia called Fragment Biotech. Give you an overview of the research program and obviously why saliva, and we all saw during the COVID-19 pandemic, in order to stop the spread of COVID-19 or the SARS-CoV infection, we needed to have field testing. Rather than going to the hospital, if you're COVID positive, you do the test at home. Because if you go to the hospital, you are going to infect others. And that's, I think, is the moment when saliva testing came to the fore. Because the traditional blood-based testing is what's currently done across the world in pathology labs. I'll also show you some of the saliva-based tests that's currently in the market, and two of the commercial products based on saliva from my lab, and applications of saliva and liquid biopsies. The program consists of four programs, and that's the oral microbiome. So that's discerning the bacteria percent. So bacterial metatranscriptomics and associating that to disease diagnosis and progression. Work on use of saliva for heart failure. Heart failure, there are over 68 million people diagnosed with heart failure. One of the unmet clinical needs is early diagnosis and prediction of patients who will require rehospitalization after hospital discharge. Working on number of cancers, and we are also developing a saliva-based test to early detect liver fibrosis, which then leads to liver cirrhosis and liver cancer. As I said in the start of my talk, 2 arms to the program, salivary diagnostics and liquid biopsies, under the umbrella of salivary diagnostics. So we do biomarker discovery, validation through to commercialization, bioassay development, and we also recently started developing biosensors with engineers both from US and from Europe. And we are also developing wearable diagnostics. So we call it a saliva denture to monitor physical and cognitive performance of Australian soldiers. And wearable diagnostics is funded by Australian Defence. Why saliva? Pretty obvious, right? Non-invasive, easy to collect. And if you ask every time we ask the patients, would you like to give saliva? Patients never say no. But if I go and ask, would you like to donate blood? Sometimes they hesitate to give blood. There is also minimal risk of contracting deadly bugs, ideal for challenging clinical environments, especially as a screening tool to pick cancer or pick diseases within a high-risk group. These are a number of saliva tests already in the market. COVID-19 test, you can buy from, I think, Walgreens in the US for $40 US. Hormone tests, drugs of abuse, saliva pregnancy test, HIV detection test, nicotine test, and I don't know whether you guys have done 23andMe or Ancestry.com to see your heritage. This is one of the products from my lab, and it's called Cancer Detect, to early detect oral and throat cancers. These are 2 major types of cancers in the head and neck area. Oral cancers are caused by excessive tobacco smoking or cigarette smoking and alcohol consumption. And throat cancers are caused by human papillomavirus. What we did is that we used the bacterial gene expression or metatranscriptomics, and then we coupled host cell gene expression, and we develop an algorithm based on AI and machine learning and develop a cutoff to determine who has cancer, who doesn't have cancer. It's what it does. And this work is published. We have 3 papers, and this work was brought to, it's commercialized by Viome, and it received FDA approval under the Breakthrough Device designation in 2021. It simply collects saliva, and there is a proprietary buffer to preserve DNA, RNA. You send the samples through to Viome, and then those samples, bacterial RNA is isolated and then goes on to next-gen sequencing, including bioinformatics pipeline, develop a machine learning AI-based algorithm, and if it's above a certain cutoff, patient has cancer. Below a certain cutoff, patient doesn't have cancer. And you can see the sensitivity and specificity. Cancer-free specificity is about 95%, sensitivity 90, throat cancer sensitivity 90. I'm just going to play you this video now. You can see it's called Cancer Detect from Viome. It comes as a box, And in the box, you get the saliva collection devices, plus FedEx. So you collect the saliva and you put it into the FedEx sample, and they come and collect the saliva sample. And then the analysis is done in the US. See the FedEx? It's just a funnel to collect a saliva. Close the lid and you send it. This is the second commercial product, and that was licensed to a startup company in Melbourne, Australia, called ESN Clear, and that's to early detect heart failure. So they have developed a device that you collect saliva sample and you touch onto a phone. It goes onto a cloud, and the cardiologist can read the results. You don't have to go to the cardiologist. So a place like Australia, large, most people live in the coastal sites, but some people also live in rural and remote communities. So this actually delivers access to healthcare because it's all on a cloud. Heart failure, by definition, is the inability of the heart to pump sufficient blood flow to meet the needs of tissues for oxygens and nutrients. You know, it becomes, I feel kind of right now like this flying donkey, because you put a lot of pressure onto the donkey and the donkey is flying. It's just like when you put a lot of pressure onto your heart, this is what happens. And the current diagnostics are ECG, echocardiogram, chest x-rays, blood tests, BNP, NT-proBNP, and all these other different tests. This is really a growing problem because worldwide 64 million adults are diagnosed with heart failure. And because of the aging and growing population, this becomes even a larger problem. A person 40 years of age has 1 in 5 lifetime risk of developing heart failure. 1 in 4 heart failure patients aged 65 years are re-hospitalized within 1 month. So think about the healthcare cost to the country. When we started off this research about a decade ago, NT-proBNP is a current natriuretic peptide currently used as a biomarker to differentiate between dyspnea versus heart failure. And our working hypothesis was that, can we detect NT-proBNP in saliva? This is the y-axis presents the NT-proBNP concentration, x-axis the patient, 45, controls 40, age match, and you can see that you can detect NT-proBNP in saliva, and with a specificity of 100% and a sensitivity of 82%. We then went and measured another marker that's used as a prognostic marker, which is a fibrosis marker, galactin-3, in saliva samples of patients and control. You can see that in the controls, low levels, patients, high levels, and this is just a Western blot to demonstrate that we are detecting galactin-3 in saliva, and there's also a correlation. And we also found that galactin-3 concentrations in the patients who have events, what we defined events as rehospitalization, death, or exacerbation due to heart failure. And you can clearly see that if patients have elevated salivary galactin-3 levels, their prognosis is poor. We then went to do a novel biomarker discovery using SWOT-MS. And this method is a data-independent acquisition, which combines the advantages of a shotgun method and a SRM, which is the selected reaction monitoring method. And using SWOT, we can discover proteins that are present in saliva. And we found, I think, over 500 proteins or peptides, and then we went and screened based on the relevance, biological relevance for the development of heart failure. We picked 6 proteins, and these are the 6 proteins. And if I go with calcarine 1 controls, these are the early stage heart failure versus late stage heart failure. Then we combine all these markers into a marker panel, and you can see in the controls, early heart failure, late heart failure. You can see we can clearly discriminate a healthy age match group from a heart failure group. C-reactive protein is got nothing to do with heart failure, but it's a marker for ischemic heart disease, and it's also a common inflammatory marker. And we were interested in, can we detect CRP in saliva? So the CRP levels in saliva of patients from ischemic heart disease is higher, significantly higher, compared to volunteers or healthy controls with a strong correlation. So in that part of the research, I've shown you that we can use saliva as a diagnostic medium to detect what happens systematically. So within the system, heart failure, circulation, and what we believe is that these markers that get secreted into blood gets secreted back into saliva, either through diffusion or filtration. There are different mechanisms these molecules enter saliva. Liver fibrosis is a huge issue, especially in Southeast Asia. Liver fibrosis lead to liver cirrhosis, then lead to liver cancer. Over half of adults over 50 years have liver fibrosis. Estimated cost in Australia is about per year 432 million. And that's predominantly because of in and out patient. Currently, what's been done is they do ultrasound, blood test, occasionally liver biopsies to diagnose liver fibrosis or liver cirrhosis. And we are proposing a saliva test because it's non-invasive, easily accessible, and anyone can collect saliva. We tested, again, the markers that are found in serum to see whether we can detect them in saliva. So serum GGT, it shows healthy liver disease, fibrosis, cirrhosis, and saliva GGT. You can see the relationship. And HA, hyaluronic acid, you can also see in serum. And also in saliva, you can use that to discriminate a healthy cohort from a fibrosis or liver cirrhosis. So is TIMP1 and A2MG. When we looked into the correlation between blood and saliva, we also see a strong correlation. As I've shown you the second application, we can use saliva to early detect liver fibrosis. That means that you can intervene early so that patients don't develop liver cirrhosis, and then the end is liver cancer. Head and neck cancers are the 7th most common cancer. These are a very nasty type of cancers. These cancers occur in the nasal cavity. These are caused by EBV virus, and these are called nasopharyngeal cancers. These are common in China. Then you have oral cavity cancers or mouth cancers caused by smoking in Sri Lanka and India, betel nut chewing, excessive alcohol. Salivary gland tumors are very rare type of head and neck cancers. The other type of head and neck cancers are oropharyngeal cancers, and these are caused by human papillomavirus. So this is the same virus that causes cervical cancer. Sadly, because of late diagnosis, only 20% of patients survive. And if it's detected early, 80% of them survive One of the other unmet clinical need is patients develop recurrences and mets within 2 years, 30% of them. HPV-associated oropharyngeal cancers, as you can see, is increasing globally, especially in the Western community compared to Southeast Asian communities. We have developed a workflow to detect high-risk HPV types that cause oropharyngeal cancers. So what we do is that we get the patient to do a swish and gargle. So we call it a oral rinse. Why we are doing this is the HPV virus resides in the tonsillar crypts. So we want to get the cells that are integrated with HPV, and the way to reach out to tonsillar crypts is through a swish and gargle. We also examine for periodontal disease because there is a direct correlation between periodontal disease and HPV infection. And then we run real-time PCR. So we've developed an assay to detect high-risk HPV types 16, 18, 33, 35 in saliva using a qPCR and a digital droplet PCR. This shows you the blue line is the HPV 16 and the orange line is the reference. So the reference doesn't change, as it shouldn't change. And you can see these are the standard curves. So we get a slope of about minus 3.43, which gives about 90% PCR efficiency, which is good. And this is a multiplex. So we started a trial before COVID, and that was funded by Johnson & Johnson because they were interested in developing a HPV therapeutic vaccine. You all know Gardasil vaccine, which is a preventative vaccine. So it doesn't help people like myself or people who have like over the age of 30. And we ran this trial at the University of Queensland Dental School, where we recruited 665 cancer-free individuals. We detected HPV 16 in 12 patients, and 3 of them were persistent. Persistent means within 2 years, if the body can't get rid of the HPV virus, you are at risk of developing HPV-driven oropharyngeal cancers. All 3 were seen by ENT guys, and they put a narrow band imaging and look for abnormalities in the head and neck region. The first person still comes, no cancer. Second one is a lady. She had a pre-malignant lesion, which were removed by an ENT surgeon. And the third one is a retired teacher who was living in Bangkok, and he said he had a very promiscuous life in the past. And for the first time, we detected a 2-millimeter occult cancer at the back of a throat of a healthy asymptomatic person. And you can see when he came to us, his HPV DNA levels were that high. And then after the tonsillectomy, person came to see the ENT guys 2 weeks later, and the HPV levels were non-existing. So he comes regularly, there is no HPV, and the guy only had a tonsillectomy, which costs about $2,000, and he did not undergo radiation or chemotherapy. So this is another study that was done by my PhD student, now a postdoc in my lab, and that was to investigate the HPV in tumor tissues and correlate that to saliva. So we had 491 patients with primary head and neck cancer. We detected high-risk HPV DNA in 81% of the patients. 50% of the patients had unknown primaries, which means that you don't know where the primary occurs. And what we found is that, and also in a separate cohort of 10 patients with recurrence, we also tested to see whether when a patient develops recurrences, does HPV come for a patient with P16 positive oropharyngeal cancers. And lo and behold, we had 6 of them to have to be P16 positive. Out of the 6, 5 of them had salivary HPV at the point of recurrence. So which means that we can use salivary HPV as a disease surveillance tool. This work all published. We then, switching gears to early detect oral cancers. And to cut a long story short, we did sequencing. We did sequencing, and then we used the TCGA database to identify microRNAs that are differentially expressed between patients and controls. And that's what you see here, patients and controls. And then we developed a panel. So what you see here is the control group. OPMD is the pre-malignant group. They are called oral potentially malignant disorders and oral cancers. So for MIR A, you can see that it increases between pre-malignant and malignant. What we then did is that we combined this into a panel. And this is what the data shows, is that we can discriminate an oral cancer patient group from healthy controls with an area under the curve of 0.96. Sensitivity, specificity is quite good. And we can also discriminate a pre-malignant lesion group from oral cancer. Then we were interested in, can we detect these markers, these microRNAs in tumor tissue? What would the expression look? So for that, we tested in oral cancer tumor tissue and healthy oral keratinocytes. You can see that it's elevated in the tumor tissue itself. We also did FISH, and you can see that in the tumor area, the microRNA is elevated. Give me 1 sec, please. And this is a busy slide. What it shows you here is the probability of developing oral cancer. So one of the unmet needs, or the physicians, what they're faced with is that when a patient has oral potentially malignant disorders, they don't know who will develop cancer, who to send home, and who to do treatment. Control group, you can see lichens planus, low-grade OPMD, high-grade OPMD. And what you can see is that these microRNA, this is one of the MIR A, I think. Excuse me. It increases. And even in stage 1, 2, 3, it goes up. And when 6 weeks prior, we had 2 patients that had high levels of microRNA, but they were categorized as oral potentially malignant patients. But 6 weeks later, they were diagnosed with cancer. So it's quite powerful that we can use these microRNA to predict which patients will develop oral cancer. We'll now switch to liquid biopsies. As I mentioned at the beginning of my talk, these are the biomarkers found in body fluids. We use circulating tumor cells, circulating tumor DNA, microRNA, exosomes, and circulating ctDNA. The rationale for using liquid biopsies is because tissue biopsies are invasive. Sometimes, especially when a patient has lung cancer, you can't access the tissue. Because of the tumor heterogeneity, you can miss relevant tumor clones. Sequential tissue biopsies in individual patients for real-time monitoring of therapy responses are less feasible in a clinical practice with tumor tissues. So the cancer management is moving towards liquid biopsies. And for lung cancer management, there are already ctDNA testing for mutations. Certain mutation, I think, such as KRAS or BRAF for melanoma. And when a patient develops resistance, you can use that to see whether a patient developed resistance or not. And the applications goes from diagnosis to prognosis to minimal residual disease detection, therapy response, resistant, relapse. Circulating tumor cells are the cells that are detached from primary and secondary sites. These are used as prognostic biomarkers for breast, prostate, colorectal, and we are now trying to say you can also use CTCs in head and neck. Among the cells that have detached, only about 0.01% of them can actually cause distant mets. And these are very rare cells, 1 to 10 cells, depending on the stage of the cancer, compared to 1 to the 10 red blood cells and 1 to the 10 to the 6 white blood cells. CTCs were first discovered in 1869 by an Australian physician called Dr. Thomas Ashworth. And what he found is that when he was looking under the microscope of a man with metastatic cancer, he saw cells identical with those of the cancer itself, and he postulated that this may shed light as to how cells give rise to metastases. And every year we celebrate Thomas Ashworth. This work, so we now want to isolate CTCs, and we use microfluidic device, and we use size exclusion. White blood cells are less than 10 microns, and CTCs are larger than 10 microns. And if I play this video, you can see small cells, white blood cells, large cells going. That's how we separate them. Once again, small cells go to waste channel, large cells go to the CTC channel. Then the way we identify a CTC is that it's a cell. So it should have nucleus, DAPI positive, pan-cytokeratin positive, which means that epithelial cell. CD45, which is a white blood cell marker, should be negative. We also expand CTCs in culture to do drug testing. This slide shows you different types of CTCs, single CTCs, CTC clusters, CTCs with white blood cells. This is a trial that started about 3-4 years ago, and it's ended, where we recruited 119 head and neck patients at baseline, treatment naive, 13th week when they come to get a PET-CT scan, and that's the time they see whether the patient has responded or not. One of the issues with PET-CT, because you get equivocal results, which means the gray zone. What do you do if you get a result like that? Currently, it's not possible to do any treatment based on an equivocal PET-CT. What the clinicians do is they ask them to come back again for a second PET-CT. So we collected blood 6 months, 12 months, 18, 24 months, and this window was used because most patients, about 20% of them, develop recurrences at 24 months. These is on the y-axis, the CTC numbers. These are the complete responders and incomplete responders. So we found 11 of the patients to be incomplete response or events. Out of those event patients, we detected 10 of the patients CTC at the baseline. So already at the baseline, you can stratify patient into low risk, medium risk, high risk based on CTCs. And we also found that CTCs at diagnosis can predict recurrence and death for up to 4 years in head and neck cancer patients. And this shows that in patients with, this is a recurrence and death. You see that patients who develop recurrences have large number of CTCs compared to those who don't. Similarly, patients who die also have large number of CTCs compared to those who don't. And if you look at the Kaplan-Meier curves, patients who respond well, baseline CTC number predict poor outcomes. Recurrence, first figure is recurrence and second figure is the overall survival, and they discriminate quite well. What we also found is that if head and neck patients who have CTCs at a follow-up time point are 2.5 times more likely to develop recurrence or die from cancer. We also culture the CTCs, and that's to do drug testing, because most of the time patients die because of side effects of drugs. What we do is that we collect the tumor tissue, we collect blood, we isolate CTCs, and we expand CTCs. One of the first questions that we wanted to address was that the CTCs that we culture, are they derived from the tumor? And in order to do that, we did whole exome sequencing, and this is shared mutations between CTC cultures and respective tumor tissue samples for 2 patients using whole exome sequencing, and you can see common mutations. This is in partnership with one of my collaborators from Peter Mac in Melbourne, where we injected CTC cultures into NSG-A2 nude mice, and the mice grew cancer. So this tells us, and the cancer was confirmed by a pathologist, P16 positivity and cytokeratin positivity, which gives us really evidence that the CTC cultured are derived from tumor cells and they can lead to cancer. We also work, this is one of my PhD student, Tilini, we also work on tumoroids. So tumoroids, what we do is that we get the tumors, we mush it up enzymatically, we sieved it, and we develop them into spheroids. And we are using this for drug testing. And you can see these are the little tumoroids. These tumoroids have different morphologies. You can see some of them look like a cyst. And we've also, how would I say, H&E stained and characterized them to be enriched of tumor cells. We then did treatment. These are treatments done in 3 tumoroids from 3 different patients using current treatment, cisplatin, paclitaxel, cisplatin, and paclitaxel. So let's look at the first patient, blue color cisplatin. These are dose response curve. Paclitaxel, green, and cisplatin and paclitaxel has a synergistic effect. Even if you look at patient 2 and patient 3, it's the same. So I've shown you that CTCs at diagnosis can predict recurrence in head and neck patients at 13 weeks. Head and neck patients who have CTCs at a follow-up time points are 2.5 times more likely to develop recurrences. And I've also shown that CTCs can be used as a model system to do drug testing. I'll quickly show you data on lung cancer, which is the leading cause of cancer-related mortality. 5% survival is 18%, and more than 1.8 million estimated new cases each year. We also counted CTCs, and you can see early stage, late stages, the CTC numbers increases, and we can also discriminate patients who have progression-free survival from those who don't have progression-free survival based on CTC numbers. So if a patient has CTC number greater than 2, poor prognosis compared to better prognosis for less than 2. We then were interested in identifying whether if a patient has an ALK rearrangement, can we pick that ALK rearrangement on the CTCs isolated from the same patient's blood? For that, we did a FISH. If there is no arrangement, you get the red and the green together. If there is an fusion or disarrangement, they separate. So you can see here, they separate. And then we use this fish, and what you see here is that in tumors that are ALK positive, we can detect ALK positivity in CTCs. If they are negative, we don't detect ALK on CTCs. How does the future look like? I think saliva and liquid biopsies will become mainstream for early detection, prognostication, disease monitoring in cancer patients. With that, I would like to thank the team and all the funding bodies, and I'm happy to take questions.
Thank you very much, Professor Chamindie, for the very interesting talk related to the industry and like industry collaborations and also the research the lab you have done. So now the floor is open for the questions. Is there any questions for Professor Chamindie? Yes, Alireza, please go ahead.
Thank you very much. That was very, very nice to hear all, and actually quite a lot of work that you have done at all the different tests. And so just wondering, because, you know, I don't know, no, I imagine Australia, the situation is different than what is happening in Europe. The IVDR and in vitro test rules and regulations did undergo changes in Europe, as you know, probably better than me, before COVID and during COVID. And so how do you deal with that? How do you somehow, with the notified bodies and so, or do you at all somehow provide your tests and so in Europe, or is it just in Australian market that you are bringing them?
So I can tell you about the Viome test. That's a FDA approved breakthrough device designation. It's only available in the US, and they are trying to get a CE mark, the company. I'm not doing any, you know, I don't do any commercialization. It's just once it goes out of my lab, it's out. But in Australia, like in the US, we can do laboratory developed test and give it as a research use only. But my lab currently is not accredited to provide services to the hospitals, even though all the clinicians are using the data that we generate in clinical decision making, for especially for patients who they have no means to gauge. So they just tell us, look, give us some clues as to what's happening with this patient. It's not a mainstream. So I'm now trying to get my lab accredited to provide official services and be paid for that.
And this process and all the things that is there, it's just research use. It is not for to be in the hands of
right now it's research, but I want to give it as a service.
Yes, Subhashini, please go ahead.
Thank you, Professor Chamindie. Sorry that I cannot show my face. I have a small technical question. If I understood correct, like in your lab, you have developed an in-house qPCR for HPV detection in head and neck cancers. So, can you tell how much would the test cost, and have you tried the same qPCR in the cervical scrapings also, and also do you use a primer-based qPCR or a probe-based one?
Okay, so there are multiple questions. So let us just start with the assay. Assay is a primer probe assay, and it's a qPCR plus dPCR. We have sequenced the amplicons, and we are quite confident that what we are detecting is the same HPV type. In addition, we've sent those samples to other companies, and they have come back validating our assay. We don't research with cervical cancer because I'm not sure whether you are aware, Gardasil, Professor Ian Fraser, Gardasil was invented in Australia. So Australia will be the first country in the world in 2035 to eradicate cervical cancer. We hardly have cervical cancer in Australia because everyone, even my son, got vaccinated when he turned 11 years.
Mm-hmm. Okay. Yeah, because in Sri Lanka, the prevalence of cervical cancer is still high, and the screening is not very strong because HPV testing is quite expensive, and that is why I asked like how much it would be how much it would cost for this qPCR, because if it is like a primer based one, it should not be much expensive then.
This is primer and probe. It's not salivary.
Okay.
So I did the calculations because the hospital wanted that. So it will cost about $100. That's excluding labor.
Okay, it's a lot, I think. Okay.
But we can talk if you are interested, because I'm also Sri Lankan. I'm happy to give back. So if you are interested, you know what's happening, you can send me an email.
Yeah, sure. Because I was studying this earlier for my master's. I was doing this self-sampling of cervical scrapings and trying to develop a PCR-based assay, and we were not very successful. That was why I was asking whether you have tried this.
We did a trial through University of Colombo because I have an adjunct professorship there, and we found that in the Sri Lankans, it's not HPV 16 like in the Caucasians, it's the HPV 18, more prevalent.
Yeah, thank you very much.
Just one more question from me. Actually, so I've seen that you have most of your research has gone to industry at the end. So are you starting to thinking that you can give this to industry, or when you do the research at certain point, you realize like, okay, this needs to go to industry. So what is your view regarding this?
So I'm a firm believer in giving back to the community. The way to give back to the community is to get industry involved and develop something because as researchers everywhere in the world, the research is funded by taxpayers. We have a duty for the taxpayer to have something out, so to improve the outcomes. I always think research needs to be translated. I've always thought from my master's time, my PhD time, even through my master's and PhD, small translation, like the essay that I developed through my PhD was embedded in the hospital in South Africa. So I'm very translational. At the start, it's just basic science. You don't know what you get, right? But then once you see good results, then you want to find partners to develop it further.
And there's one more, like I have seen that there's one patent for you in your research team. So like from the start to the patent, how long it took to get done the things? Roughly, like how many years, or like how many PhD
I can
students?
give you, one took 5 years, 7 years, and the other one took 3.5 years with my PhD student. So it takes a long time. It just doesn't happen just like that. Yeah. But for me, I worked in industry. I used to work at Philips Electronics in Netherlands. So I'm Dutch, I'm European. Well, I don't look European, but I hold a Dutch passport. So I lived in Netherlands for 10 years. And I come with my industry background into academia. So I have that skill set already.
Okay. Yeah, I understand. And like probably the last question is like, how challenging the life as a female scientist to deal with both the academia and the industry?
Oh my God. I don't know how many of you guys are online. I don't want to scare anyone. It's not for the faint-hearted. I can tell you, when I started as a postdoc moving to Australia, I used to bring my 3.5-year-old son, put him in the office and go into the lab on weekends and work to generate data to get a fellowship. So it's been ever so tough, especially in Australia, because there's still not many women in academia or in high positions. And especially if you are a woman of color, then the issue is even more. It's completely different to Netherlands, where it's equal. They don't care whether you are a female or a male, provided you do the job, you get the job. There's very subtle bias here.
Thank you very much. And if there's no more questions, I would like to say thank you very much for your time. And once again, so sorry for the timing issue.
It's all good. It's
And
5:20 on a Friday afternoon, so I'm about to go home.
Happy Friday, and thank you very much.
Thanks.
And we'd like to see you someday in Estonia too.
Estonia. I have never been to Estonia, so yes.
You're very welcome. Yeah.
Thanks.
Thank you very much. See
Take
you.
care.
Thank
Bye.
you. Bye.